So in the today's presentation, we will be discussing mainly about the forensic food fraud analysis. So it is having two components. One is a forensic component, another is called a fraud analysis component. So we know that the demand for the meat, the price, everything is increasing. So we have more than 70% of non-vegetarian population in our country. And the demand for ready-to-eat and ready-to-cook meat products are increasing day by day, which is due to changing the preferences, consumer preferences, and which is changing due to the purchasing power. So with this high demand for animal-derived products, maybe due to the preferences, due to palatability, due to nutritional requirement, these commodities are always prone to adulteration. So we will be discussing about how the forensic component is coming into this meat authentication and how the food fraud issues can be tackled. So all this has been covered under three different areas called as, one is called as the species identification, another is called as the sex identification, another issue is called as the tissue identification. So tissue identification we will not be discussing during this presentation, because as already Vaidyanathan sir explained, the tissue identification is required when we talk about the organs or the offal meat. So we are not discussing that part during this presentation. We will be restricting our discussion to the species identification mainly and the sex identification. So now, if we see the food fraud is having many forms. The food fraud, especially the meat fraud, can be an adulteration, or it can be a substitution. So substitution is replacing a costly meat with a cheaper meat is called substitution. Then it can be a dilution, it can be tampering, counterfeiting, and mislabeling. So there are 7 to 8 different forms of food frauds exist. So, but in today's presentation or today's interaction, we will be mainly concentrating on 2 forms of food frauds, which is called as adulteration, or per se the substitution. Another is called mislabeling. So mislabeling is nothing but labeling one animal species with other species name. That means a cheaper species it will be labeled as a costly meat. And another is called as the substitution, or adulteration, where one part of a costly meat being taken out, which is being replaced by a cheaper quality meat. So, if you see the food fraud in globally, so to mitigate the food fraud in the global scenario, we have 3 different approaches. So the first, let's see, there is a chance of an unintentional adulteration. So unintentional adulteration, for example, there was an issue from Malaysia, where traces of the pork DNA were detected in some of their meat products. So that is a concern, especially in the halal point of view. So that is not an intentional substitution that happens due to a contamination. That means from the raw materials, from the raw material contamination or the processing environment or the processing machinery. So such forms of unintentional adulteration or unintentional form of contamination, we can tackle by following the principles of food safety or by following the principles of hazard analysis critical control point. So that is a very basic concept. But now the latest concept in this area, that is called as food defense and food fraud mitigation. So you please give some attention to this area. This is a very new concept in food fraud evaluation. So we have intentional adulteration by 2 methods. The intentional adulteration can be an ideologically motivated, or intentional adulteration can be economically motivated. So you see that there is a great chance for ideologically motivated issues, ideologically motivated to defame some company, ideologically motivated using the social medias to take revenge against some company by their own staff, their own previous staff. So these are called as the ideologically motivated issue. So ideologically motivated issues are discussing as a food defense issue, and which is being tackled by a concept called as TACCP, that is the threat assessment. So that is a threat. So, but in a food safety point of view, we are more interested to tackle the issues which are economically motivated. So such economically motivated adulterations are being tackled by food fraud mitigations are tackled by a new concept called as VACCP, that is the Vulnerability Assessment Critical Control Point. So today's presentation and today's discussion, we will be mainly discussing all these economically motivated adulteration. So let's see, from the laboratory's point of view, we have many customer base, and we have related to many of the regulatory bodies. So the kind of the regulatory bodies will decide what is the kind of the requirement, what is the test has to be performed. So you see, in most of if you see the slaughter of the forbidden animal species or the unconventional meat, you see in our country, many of the states are having some form of restriction on the slaughter of the animal, except the northeastern state and the Kerala and the West Bengal. So the requirement of animal species identification vary from state to state. If you take the case of Telangana, or if you take the case of Andhra, what is the requirement? Which animal species is permitted or not permitted, which vary from Karnataka. So now the latest Karnataka, the latest issue, that even buffalo slaughter is banned. So once that act is implemented, now we are getting many samples from Karnataka to test even the buffalo species. Previously, it was restricted to the cattle species identification. So another issue is called as the unconventional meat. The unconventional meat issue, mainly as per the government of India's declaration, we have many food animals. We have many food animals, but there are certain animals which are not even, that is not declared as a food animal. So one such example is a camel. So camel cannot be slaughtered for the purpose of food in India, but we have many requests from the field veterinarians to test the meat for the presence of the camel meat. So another issue which we are facing is poaching. So poaching is nothing but wild animal, hunting of the wild animal or game meat. So now after this pandemic, even this pandemic is also is linked to the wild animal markets in some of our neighboring countries. So now the latest OIE guidelines, there should be strict monitoring of wild animal markets where there are unconventional animals, or the wild animals are being used for the food purpose. So you may be remembering the first case from India where the Salman Khan's issue, so which is being that is one of the first case from our country where the forensic has been used in animal species identification, which was not performed by NRC meat, it was performed by CCMB. So we have many samples from wild, the forest departments. So that is another issue. So now the most important and the most common form of issue what we are facing is called as economically motivated adulteration. That is the most common form of food fraud existing in our country. So this is mainly coming under the food safety department. So food safety departments are constantly they are collecting samples like processed meat products like halim, biryani. So we have many cases of evaluating biryani. Just few days before, even we received a biryani sample from Andhra, Gundur, and we have collected many samples of comminuted meat products. So this is nothing but economically motivated adulteration, as I already explained in the initial slides. This is a substitution or an adulteration where a costly meat, for example, mutton, being replaced by a less costly meat. A part of that preparation, if they are planning for a 10 kilogram of preparation, maybe 2 to 3 kilogram, they will be replacing with a cheaper meat. Usually chicken is the most common form of substituent species they are using. So another issue we receive samples is mixing of the bovine derived materials. So this is mainly connected to the import and the export clearance. So some of our colleagues are working on the animal quarantine and the certification services. That is a part of the port, especially airport. So when any feed derived materials or the health supplements, vitamin supplements, which we are importing from another country. So it has to be tested for the absence of animal derived material, and bovine derived materials is very important in terms of the import and export risk assessment, especially connected to the bovine spongiform encephalopathy. So we do receive samples in this area to test whether any kind of animal derived material is present or absent. So another area which we are getting sample is the halal authentication. So halal authentication, we won't be, we are not following the, we are not supposed to give the authentication. We will be testing the end product, that is called as a halal compliance rather than halal authentication. The test will be for giving a halal compliance. So the initial process of halal authentication will be taken care by the municipal bodies or the religious Muslim religious body. The final product testing for the absence of the pork DNA, we will be doing the real-time based testing to give the halal compliance. Then another area which we are getting sample is called the meat export. So we have export guidelines, and we can export only buffalo meat. Among the large ruminants, we can export only buffalo meat, deboned and deglanded meat. So that is one of the main area we are getting n number of samples, because there is a chance of other bovine meat maybe exporting. So there is always a vigilance from the police department. So there is always a requirement from the police department and the export development bodies like APIDA to evaluate the sample. All these are the regulatory requirements. So in a meat plant, we may evaluate a meat species based on anatomical methods or based on the experience. But whatever the area, what we are discussing during this presentation is all are regulatory requirements. So regulatory requirements need a robust test method, and the regulatory requirements need a test that has to be performed in an accredited laboratory. So here comes the importance of NABL accreditation. So now in the 2019, you see in India, we don't have any Indian standard or the BIS standard for animal species identification. But in the year 2019, the ISO has come up with a standard called ISO 20813, which is an excellent standard. After the release of this standard, now the laboratory can easily implement any of these analytical methods in their laboratory to produce such a legally acceptable report or animal species identification report for regulatory bodies. So now this is just to give an idea about the meat adulteration issue globally. So not only in India, this is a global issue, the meat adulteration issues, because the horse meat scandal, which was happened in 2013. So that was one thing that opened up the eyes. And even though in 1981, we have the Australian great meat substitution scandal. So there are n number of issues like Florida fish scandal. So globally also, this meat adulteration is a very big concern. But the comparison to our country, globally, that is mainly dealing with the economically motivated adulteration. But in our country, mostly it is connected to the cow slaughter issue or the wildlife animal meat issues. So, but now slowly, our regulatory requirement is becoming more stringent, and the adulteration issues are also coming into picture. So now there are certain reports which shows that the considerable quantities of pet food are being illegally diverted with the human food chain. You please understand what is the issue, and there is a great decline in the requirement of the offal. So what happens to these offals when there is a great decline in the requirement of the offal? Finally, these offals will end up in any of the meat products like processed meat products. So we have to evaluate all this scenario in terms of public protection. We have to see the public health point of view, and we have to protect our industry, and we have to improve the trade of animal-related products. So kindly see, as I explained, the regulatory body will vary, the type of the sample will vary. So we have many different samples. So we received the horns from various states. So sometimes we receive the carcass meat Usually that is the meat. The meat, usually the veterinarians are sending meat. So sometimes we receive the bone or any horn or any byproducts. So when it is related to the food safety department, they are sending cooked meat products. Sometimes we are receiving the nowadays we are receiving the samples from the police department, the blood smears. We started getting samples called as blood smears. Then sometimes we are getting the slaughterhouse waste, sometimes unidentified tissues. So the sample vary. The most important issue is the heterogeneity. The sample vary. There is no constant form of sample. There is no preservation for the sample. So after this interaction, at least few of you may come up with good sampling procedures for sending the samples to such laboratories. So now this is just a slide to give you a brief idea about how the regulatory sampling exists. So we don't have any sampling. Usually the animal husbandry department for this particular test, we don't have any sampling plans or sampling procedure. But for the food safety department, they have a very robust sampling. So in every case or any case, they will be collecting 4 samples from that one investigation. So the food safety officer will be collecting 4 samples. And out of that 4, the 1 sample, they will be sending to the state food safety department. So we have the state food safety laboratory in Nacharam, Telangana. So out of that 4, 1 will be going to the state food laboratory. So another 3, the 2 will be with the designated officer of that district. So then if there is a requirement or a request from the hotel person or the food business operative, they are called as food business operative. The 4th sample they can send to any of the NABL accredited private laboratories, not to NRC meet or any referral laboratory, to a private laboratory. So now 2 samples are sent to for analysis. 1 sample to the government laboratory, that is the state laboratory, another sample to an NABL accredited laboratory. So if there is a contradiction between the report which is produced by the state laboratory or a contradiction between the report produced by the NABL accredited private laboratory, then comes the strategic role of referral laboratory. So now the 3rd sample, the designated officer will be sending to the referral laboratory. So NRC meet is the referral laboratory for this animal species identification for the entire country. So just remember, dear colleagues, since we don't have any sampling framework, at least I would like to explain one of the sampling procedure that is adopted by the food safety department. So this is a sampling plan, or that is a regulatory requirement. So when it comes to the veterinary department, in all these cases, let it be a wildlife case, let it be an animal husbandry related or export or import, ultimately the veterinarian of that area, the veterinary doctor will be involving in the sample collection. So that is a very important aspect of the testing. So you have to collect a very proper sample. If it is a meat, you have to take a 50 to 100 gram piece. Or if it is a meat product, you have to take the entire unit of that meat product. So so now you please see, once you have a very good sample with you. So yesterday I have received a larger femur, an entire femur from one of the animal husbandry department. So you see, if you have a meat sample, you please send that sample to the laboratory. If you are sending a femur sample, the things are very difficult from our side to extract DNA from such samples. So in a nutshell, dear doctors, you please try to collect a 50 to 100 gram of meat samples and take the very simple form of preservation. So one of the major issue what I'm facing is the use of formalin. So we used the formalin in n number of cases during our UG studies, or formalin which is using by the food safety department. But kindly see, if we are interested for any type of molecular analysis, if we are interested to extract DNA or RNA and to perform a PCR, kindly please avoid formalin. So the very simple form of sample preservation is salt. So you can take a meat or a meat product, you can put salt into a plastic container, you can place your meat over the salt, then you can close that container by adding much more salt, then you can go for sealing with the Panchnama. So kindly see that when the salt is the best option, that will protect the sample for maybe 7 to 10 days. There won't be any issue. A washing will remove the salt and I can extract best quality DNA. So formalin, never use formalin. And if you use the normal saline, that is another common practice, the normal saline. The normal saline will not take care of any preservation. The meat will be, once it is reaching to Hyderabad, it will be highly putrefied. So one of the message from this lecture, please take 50 to 100 gram of meat and pack it using the salt. So these are some of the samples we are receiving. The tallow. So you know that what is a tallow, which is a rendered animal fat from a ruminant, and the lard, which is a rendered animal fat from the pork. So this component is of great importance in the context of the adulteration of ghee. So there is a probability that the ghee from cow or the ghee from buffalo may be adulterated or can be adulterated with the tallow from the same species. So we have identified many of such cases. So we receive samples on tallow, and in the center we have an unidentified bone. So this was a very special case. We received a sample from Kerala. So even though the bone looks a little bigger in this image, it was very small, maybe one centimeter or something like that. So this bone was thought to be from dog. So there was a social activist. He created hiccup in the state of Kerala. So finally, the Kerala state sent the samples to the referral laboratory, and we have identified this bone as a bone piece from cattle. That was a cow bone, but as per the state regulation from Kerala, that was not an issue. So then we have meat curry from wild animal. This was caught by the forest while people were hunting animals. They prepared the curry and they were consuming in the forest. So the forest department caught them. They sent this curry to NRC meat for identification. Now gradually what is happening, from a regulation, from food fraud, the things are moving towards a more forensic side. The police is involved in many cases. One such case is a metallic object. So there was a controversy happened in Punjab where one person has stolen one of the animal from his neighbor, and he cut the animal and he cooked it and consumed it. So there was a case, there was involvement of media and all those things. So they want to identify the real culprit. So they have seized certain metallic objects that they used for cutting the animal, for digging the remnants and all. So the police is coming with this kind of samples to NRC meat nowadays. So what I want to say, from a food fraud, it is going to a forensic side. So in all these cases, dear friends, in all these cases, the veterinarian will have to play a very important role in collecting the best sample and collecting the most suitable sample. So in one case, we have received a putrefied meat. So you see, this is the issue when we use a normal saline. So the doctor has added a normal saline once we received at Hyderabad. This was the condition. So extracting DNA from this sample seems to be very, very difficult. So, so now I hope you have a very broad idea about what are all the regulatory requirements, what are all the regulatory bodies, what are all the different types of samples that can be evaluated. So now you see this process is different from usual veterinary evaluation methods, veterinary laboratory techniques. The main reason is that it is a legal process. So far we have evaluated, myself and Dr. S. Vaidyanathan sir, we have evaluated almost 415 samples. So all these 415 samples are legally identified. That means one FIR is connected to all these samples when it is handled by the police department or the forest department, or where there is a legal involvement when it is handled by the food safety department. So we must say that since it is a legally bound activity, so in some of the state, the bail will not be granted until the report reaches to their department from NRC. So such a kind of complexity is evolving. So now since it is legally involved, the result should be accurate, So the result should be unambiguous. So that is another important aspect. Whatever we do, the test should be accurate. We have to justify our act. So whatever result we produce, it is having a great impact. So the result should be accurate. Now, I already explained, you see the kind of sample what we are obtaining, it is highly heterogeneous. That already you are all understood how heterogeneous is the sample, and the quantity of the sample may vary. So sometimes, very interestingly, I may not be able to recover a 1 gram of sample from a sample which is a packet which is sent by the department, or sometimes I may receive 20 to 25 kilogram of meat where I need hardly 50 gram, and finally I need only 25 50 milligram. So you see, this is a complex situation. So now, considering all these point of view, so the samples are heterogeneous, we need accurate and unambiguous result. So after this ISO standard, which is 20813, which I already explained, most of the regulatory laboratories, let it be in India or in the globally, all are following DNA-based methods. So we know that it starts with the post-mortem inspection called meat inspection. Then the protein-based methods came into picture. Then only the DNA-based methods came into picture. So there are n number of methods. Let it be a lateral flow assay, let it be a protein-based assay, or whatever it is. But DNA-based assays are mainly used by the laboratories around the globe. So DNA-based methods, you know why the DNA-based methods. So you know, now we have gone through n number of samples. Some are highly putrefied, some are cooked meat products. So the beauty of the DNA is that we can say that DNA is almost extremely stable. Even with the different processing conditions, the DNA remains stable. And the DNA maintain a conserved nature. So that conserved nature will be utilizing to evaluate the animal species. And the tests which are based on the DNA, which are highly specific, which are highly accurate, which are reliable, and highly reproducible. So accuracy, specificity, reliability will lead to a legal acceptability to DNA-based methods. So now, after this introduction, we will be moving to the sheep and goat case studies. And during this case study evaluation, I will be explaining the biomolecular analysis. That means someone of you may be interested to understand something about the PCR-based techniques. So we will go through some of the PCR-based techniques, and we will restrict our discussion to the sheep and goat meat. So you all are familiar that the Asian countries with the highest goat and sheep population, there is a huge demand for the sheep and goat meat in our country, and there is always economically motivated adulteration, replacing the sheep meat by any cheaper meat, replacing the goat meat with any other cheaper meat, replacing sheep meat with goat meat or goat meat with sheep meat. It varies from case to case and from the smartness of the fraudster. So now all these things demands we must follow a very strong and very robust authentication methods. So when we come to the technique per se, this laboratory performed the species identification, the sex identification, and DNA microarray. So this is an introductory slide. I will be explaining one by one. So now I all welcome you to this lab. So once the sample received the lab, the most challenging part of this evaluation is the DNA extraction. So now you have seen already, we have bone, we have putrefied meat, and we have sometimes not the note which is sent by Reserve Bank of India. We have papers, we have biryani, we have halim, and number of products are coming to NRC meat. So the DNA extraction is a very robust, very difficult part. So we have standardized the DNA extraction from all these. So yesterday we have received a bone. So bone, if it is a fresh bone, we will be definitely looking for a bone marrow. And if the bone is not fresh, we have to make a powder out of the bone and to extract the DNA. So the most challenging part is the DNA extraction. So now, I invite your attention, very keen attention for another 5 minutes. I will be explaining the test which is required for a very particular case. So if you see, when you are sending a sample to NRC meat, and you are not giving any clue to me. You are sending a sample to a laboratory and you are not giving any clue. There is no clue means I have to find out what is the species which you have sent to me. So when there is no clue, it is called as an untargeted approach. The untargeted approach is, it is a blanket study. I don't know what is the species. It may be any of the food animals, it may be cattle, it may be buffalo, or it can be a wild animal, or it can be a synthetic material. We don't know. So in such case, the technique what we performed is called as a universal primer amplification. So this is the core of our testing activity. I request your attention for 5 to 10 minutes, very keen attention. So this is called as an untargeted approach. The untargeted approach is performing by a universal primer amplification. So universal primer amplification is nothing but you are extracting a DNA from the sample. You have a DNA with you. Now you are using a universal set of primers. The universal set of primers will amplify any DNA, let it be cattle, let it be buffalo, let it be wildlife. After amplification, we will be getting a PCR product. That PCR product you are evaluating in a gel, and once you confirm the PCR product is there, that product you will be evaluating using the Sanger sequencing. So now the beauty of this is that any number of animal species may produce a PCR product. We will sequence the PCR product using a Sanger sequencing, and once the PCR product is ready, once the sequencing details are ready, this will be blasted using NCBI, and we will find out what is the sample which is present, what is the animal species that is present in the sample. So the major activity is called as an untargeted approach, which is performed by a universal PCR amplification, followed by Sanger sequencing and blast evaluation. So you please listen. This kind of test or this kind of universal primer amplification is applicable only if we have a single piece of meat. The meat should be single piece, and that single piece of meat should be from a single animal. So you see, if there is a single animal, when you are visiting a slaughterhouse, when you are encountering an issue, there is a single species or a single carcass. From the carcass, you are collecting only one piece of the meat. When you are sending that piece to Hyderabad, then we will be using this technique. So if you have, now you see when you are seizing a lorry where you have 50 packets of meat. So if you send one piece of meat from one packet, that will represent only that packet. Another 49 packets are remain unsampled. So please think all those while you are collecting the sample. So now another approach is just a minute. Another approach is that is called as a targeted approach. The targeted approach is nothing but the customer is asking the laboratory to find out whether it is cattle or whether it is buffalo or whether it is any wild animal. They are posing a question to us. So when they are posing a question to us, we are very confident that what we have to test. That is called as a targeted approach. The targeted approach, we are using species-specific amplification. So now the sex amplification is required only if we're dealing with the cattle and cattle cases where it has to be evaluated for cow and bull. So probably some of you may be thinking or maybe identified this sex identification. Sometimes it may be required in case of sheep and goat because of the changing preference towards the male or female meat. Now, DNA microarray or chipron analysis that I will explain along with the case studies. So now this is just to give an understanding. So now you see the laboratory is having a general requirements and structural requirements. So forget about those things. So if a sample is coming to NRC meet and NRC meet is producing a report, we have to control the resource, we have to control the process, and we have to control the management requirements. So you see that the process requirements are entirely dealing with the testing method. So whatever the test we perform in our laboratory, we are maintaining an internal quality system as well as we are following an external quality system so that we ensure that every result which is produced by this laboratory is legally acceptable. So this is a concept coming under NABL. So probably this NABL related things. If there is an interest, we can discuss in the future. So now I'm moving on. So this universal primer I have already explained. So those who are interested in PCR, please give some attention here. We know that the mitochondrial DNA, which are being largely used for the animal species identification. So mitochondrial DNA, you know, it is maternally inherited. So since it is maternally inherited, there is no recombination. So the sequence are conserved and the sequence will remain from generation to generation. So we have the universal primers, which are designed against the mitochondrial DNA, 12S rRNA. Then we have the mitochondrial cytochrome B. We have the cytochrome oxidase subunit 1. So we have many number of primers can be designed against the mitochondrial DNA. So once you design a primer against a mitochondrial DNA, what will happen? We will develop a forward primer and a reverse primer, as I already explained. So this is a technique even you can follow in some of your laboratory, where you can take the help of a sequencing facility. So Hyderabad, we have many sequencing facility. They may charge 400 or 500 or 600 rupees only per sample. So you can extract a DNA. You can perform a universal PCR using this mitochondrial DNA, and the PCR product can be sent for sequence. So once the product is sequenced, the company will send you the sequence in your Gmail or email. So that sequence can be used for blasting, and you can find out the animal species. So why you please, when you are planning for any of, let it be a thesis work or a laboratory test development, you please design, if you are interested for a testing or detection, you please design gene from a mitochondria, because mitochondrial genes are highly conserved and you will get best results when you work with mitochondrial DNA. Now, now we will discuss a little about the species specific PCR. So now that we will restrict our discussion to sheep and goat. So one of the samples we have received in 2016 from one of the veterinary official from GHMC. So some of you may be remembering that was a dog biryani issue. So that was again, as I initially explained, that was ideologically motivated issue. That means social media were used to take some kind of revenge against some hotels or something like that. So we received the sample. So when we receive a sample, for a dog meat issue, so first we have to test whether that is dog meat or not. So for dog meat or not, we have the dog specific or dog species specific PCR. So when you perform a dog species specific PCR, when you are not getting a result, then the next question, if it is not dog, then what it be? This is the major challenge of a laboratory. So when it is identified as not dog, then we have to identify what it is. Then after n number of studies, we have found out, we have performed the sheep and goat species specific PCR, and that biryani was identified to be mutton, that was identified to be from sheep. So this is one case study where the species specific PCR has been used. Now I already explained sequencing. That is another technique what we are using. So that is using the universal primer technique. So we have a sequencing facility. So we will be performing the entire gene sequencing. So we have the 12S gene, the mitochondrial cytochrome gene, the cytochrome oxidase unit gene. So when you are interested to perform a test, no need to design a primer. These primers are available. Even you can take from any of the NRC meet publication, or you can take from any of the peer-reviewed publication which is available. So you extract the DNA, perform a PCR, you can send that to sequencing. Even a few of your sample can be sent to NRC meet. We can help you in sequencing. And once the sequence is ready, it can be used for blasting. So this is about the universal primer technique. So now, very lately, considering the requirement of the sheep and goat meat species differentiation, our laboratory has developed a duplex PCR for the simultaneous detection of sheep and goat. So duplex PCR is nothing but you are targeting two targets at a time. You will be having a common forward primer. That primer will be specific both to sheep and goat. Then you will be having two reverse primers. The reverse primers will be species specific. So there will be one sheep specific reverse primer and there will be one goat specific reverse primer. So end of that reaction, we will be having 3 primers, and this primer has been standardized in NRC meat. Now this is under a publication stage. So we have developed a touchdown based PCR. The touchdown PCR is nothing but it's a relatively easy and new concept. Like the PCR will be starting with a high temperature where only the specific genes, like if the sheep DNA is there, the sheep will be amplifying, and if the goat DNA is there, goat will be amplifying. Then immediately we will reduce the temperature so that any non-specific amplification, means amplification from another species, will not be happening. So this is a new protocol. So now we are using this touchdown protocol for the sheep and goat meat issues which are coming to NRC meat. So now based on this, we have evaluated few of the samples collected from nearby areas of Hyderabad. So we have evaluated many samples, and sometimes it may be labeled as sheep, it may be goat, sometimes which is labeled as goat, it may be sheep. So there is a high level of mislabeling in all the sheep and goat samples, and even we have evaluated for the sex of a sheep and goat. So there is high level of mislabeling. I can say that mislabeling is very common in this species of sheep and goat. Then we have developed the PCR based techniques where to detect the presence of the chicken in sheep meat mixtures. One of our major research study, what we have conducted, is detection of multiple animal species. So you all are familiar, maybe better than me, regarding this Halim meat product, which is a highly comminuted meat product. So we have evaluated 100 Halim samples, the 50 samples from 2016 and 50 samples from 2017. So Halim samples, some may be labeled as mutton Halim, some may be as chicken or beef. So we have evaluated these samples based on a DNA technique called as DNA microarray. So I will explain a little about this. So far we have explained the cases where only one species is involved. Let it be a targeted approach or let it be an untargeted approach, only one species is involved. Now the scenario is changing. Now we can expect multiple species in one meat product. The typical example of economically motivated adulteration, where the product is labeled as mutton, some portion of the mutton may be replaced with the chicken, or some may be replaced with the beef, or in sometimes it may be labeled with the camel. So to evaluate that, we have a very special technique called as the DNA macroarray. We will extract the DNA from the product. We will perform a PCR called as a universal PCR. Then, as I already explained, if there is a DNA, there will be a PCR product. If there is one species, there will be one product. If there are two species, there will be two products. If there are three species, there will be three products. So once the product will be ready, then we are hybridizing that product into a chip. You can see a chip here where it is marked in red colors and the green colors. So this 25 represent 2 to 25 represent 24 animal species. So 24 animal species probes are embedded here. The number 2 in duplicate represent maybe cattle, 3 for buffalo, 4 for sheep, 5 for goat, chicken, like that. Every animal species probes are embedded here. When you are pouring the PCR product into this, you will get multiple points if there are multiple species. If there is only one species, we will get only 1 point. So this is one of the Halim sample which we have collected. So here you can see there are 4 red lines. The 4 red lines represents 4 species. If you see the cattle is present with a value of 61,000. The sheep is present with a value of 61,000. Goat is present with a value of 58,000. And buffalo is present with a value of 62,000. So literally we can say that 60, 61,000, 58,000 all are equal. So this is a scenario. There is one Halim product has been evaluated. After evaluation, we found out the product contained equal proportion of cattle, sheep, goat, and buffalo. So now you imagine how the mislabeling is happening. I cannot interpret whether it is a goat adulterated with a sheep or cattle or something like that. I am not ready to take that risk. But you see the end product is having equal proportion from cattle, buffalo, sheep, and goat. So this was one of the study we conducted. This is performed using the DNA microarray. So when you send any sample where you are suspecting multiple species, still we are following this technique to produce a report to the customer. So you can see now we have many Halim samples are here, like one is having, if you see the first one, we are having sheep and goat, but a very less amount of cattle. You see the value part. The first square, if you see the goat 47,000 and sheep 47,000 and goat 58,000. You see these values are equal. That means the sheep and goat meats are invariably, it is mixing, but the cattle value remains very low. The cattle value remains very low means that can be a contamination, whether the same vessels has been used or DNA from ghee, something we can expect. When the proportions are nearly equal, we can say that that is a substitution where one species is being substituted by another species. So this is an unpublished data. So we are going to publish it. So we have evaluated 100 samples including 2016 and 2017, and many samples were found to be adulterated. In 2016, 12 muttons were found to be adulterated or mislabeled. In 2017, 17 samples were found to be mislabeled or adulterated from mutton halim itself. So in beef also, adulteration were detected in 9 to 7 samples. So we can say that chicken is one of the most common form of adulterant they are using in mutton and beef product. That is to make the price compatible. So this is an unpublished data. And now this is what we have explained is simple PCR based techniques. That is for universal primer amplification followed by sequencing, species specific PCR, and the chipron analysis. Now, if any one of you are interested to go for a species identification, now no primer designing, no standardization, nothing is required because the ISO standards are available. If you see the ISO standard 20224/2020, that is for the real-time PCR based test for the ovine DNA detection, and ISO 20224 part 5. So you can directly purchase this ISO standard from the BIS office or from the ISO website. You can use these primers to design your test in your laboratory, but you need a real-time PCR. This is one of the forensic study. It was an interesting study. We have received a few samples from one of the online meat marketing. So now the online meat marketing is becoming more and more common. So we have received 3 samples from one of the online meat markets from one of the southern state of India. I'm not disclosing any of such details. Only for your curiosity, I'm including these things. So the sample, which is they sent 3 samples, which is one is labeled as mutton chops, another is labeled as mutton leg, another one is labeled as mutton shoulder. So there was some kind of confusion by the owners. So they are sourcing the meat from the northern state, they are transporting it to the southern state, and they are doing the online market. So they send the sample to NRC meat. We performed the analysis. We performed the species specific PCR. We performed all those analysis, and we found that all those meats were from pork. So you see how the fabrication of the meat is being performed to mimic the pork into a mutton meat. So this was one of our study. And after that, what that entrepreneur did, he closed his contract with the northern North Indian dealer after this result. So this is the way the laboratory is helping entrepreneurs, the way we are helping the regulators to tackle the issues. So there is another case study which is related to the sheep and goat. This is from another northern state of India that I have already explained during the Diwali time. One person at night, he wanted to eat mutton, and there was no mutton available during that time. So he went to his neighbor, he cut the animal, he prepared the meat, and he ate. But that created a big issue the next day, because animal was very emotionally connected to the owner. The owner was very emotionally connected to the animal. It became a police case. So the police finally seized the weapon they used to cut the animal. You see that this kind of evaluation were happening in animal forensic, but in the sorry, happening in the human forensic, but animal forensic, these are very new experience and very new issue. But the very important difference between the human forensic and animal forensic is that the human forensic is mainly dealing with individual identification, but in the animal forensic, we are dealing with the species identification. So this is one of the case study we performed for one of the North Indian state department. So now we are moving everything into the third generation. First we started with the normal PCR. Now we moved on to the real-time PCR, and finally now we are moving on to the droplet digital PCR. So now, as per the cases we explained, we are all interested in detection. So now, when you discuss about the economically motivated adulteration, when there is a substitution happening, or when there is a mislabeling happening, we have to quantify what is the level of mislabeling happening, or what is the level of substitution happening. So in that cases where you are interested to quantify, we have to use the very latest technique that is called as the digital droplet PCR. So we have digital droplet PCR, and currently we are standardizing the techniques for quantify. That means if there is a substitution, of chicken into mutton halim, initially now we are producing a report, what is the species which are present in that halim. So now in the coming years, we are planning, and the regulatory requirement will also be changing. What could be, or what is the level of that adulteration? So to quantify that adulteration, we need very latest techniques. So we are using the ddPCR for that. So this one or two slide I have included. There was some question during previous training program to how to go for anatomical differentiation of the sheep and goat. So that this point can be taken from the Wilson's practical meat inspection textbook. So if anybody is interested, can take a screenshot or a photograph of this slide. So I'm not going to explain because I'm not following any of these principles. I'm following only the DNA-based techniques to produce the report.